When I was applying for jobs, I got my offer from UCSD, but before I got my offer from UCSD, I got rejected from some other places. And sometimes it's nice if you get feedback from a job that you didn't get successfully. And one of the senior faculty members at one of the schools where I did not get an offer said that your proposals were regarded as a little too safe, and that we would hope that there would be a pie in the sky kind of proposal that would be high risk but potentially high reward. So I don't know how high risk this project area is, but it's really been something that I've started to think about after getting tenure. So now that I can't be fired within reason, what is it that I would want to sort of make it as a unique contribution to the field of nanomaterials, and maybe connecting some other disciplines that may be kind of disparate. So this is organic haptics. So haptic technologies, has anyone seen Ready Player One or read the book? Okay, so the haptic suit, it's like haptic technologies are technologies that interface with the sense of touch. Not just the sense of touch, but also the sense of motion, sense of kinesthesia, which is the kinesthetic sense, the sense of motion of limbs and also the physical resistance of objects in the natural world. And it is my group's hypothesis that if you can develop materials that more accurately reproduce sensations of objects in the natural world, then you can do things like create VR and AR environments for training, for surgical training, telerobotic surgery, physical therapy, devices for accessibility, for example, for neurologically impaired patients, for tactile therapy for premature infants. And also, if you have this new toolkit, it might also allow you to understand the peripheral nervous system much better than you can with typical devices that interact with the tactile sense, like motors, like vibrotactile motors that might be in a video game controller, or also maybe a phone. In fact, this slide advancer is a haptic device because when I have 5 minutes left, it shakes. Human-machine interfaces. So where are we? By means of setting this up, I would say that human culture is replete with artifacts that interact with, that engage with the senses, with the five senses, to convey information or convey a thoughtful response. So with the sense of sight, we have painting, sculpture, film, architecture, typography, as Steve Jobs continuously reminded us of, and clothing. For hearing, we have music, poetry, theater, speech, ringtones, games. These lists could go on forever. For taste and smell, I'll just combine them because often they are quite linked. We have food, beer, wine, cosmetics, perfume. But for the tactile sense, I would argue that in terms of artifacts, that is, physical objects and manifestations of culture that involve some physical embodiment, we are a little bit behind the other senses. So for example, you might think, well, massage is a good example of something that interacts with the tactile sense, and that's true, but I wouldn't say that that's an artifact of culture. It's not. It's a cultural. It's a cultural element, but it's not an object. A massage chair is, but massage chairs are kind of crappy. So I would put a question mark here. And we have things like fabrics and textiles, which of course might convey a thoughtful or emotional response. You know, we'd rather pet a cat than rub our hands on sandpaper or scratch a chalkboard. But these aren't really dynamically reconfigurable in the sense that the sound a violin makes is, or the sense that a projector can play a movie on a screen. So I would still say that this is kind of an underdeveloped area. So what I think that this might be an area to combine or to create a new interface between material science and even the humanities. So we might be able to access new forms of thought-provoking artwork or human-machine interfaces, or at least understand the peripheral nervous system better using these tools. And the analogy that I would use is to the red, green, and blue pixels in a display. So in a projector like this, we have red, green, and blue pixels that combine and trigger the cone cells in our eyes to perceive different colors, and we can get any color in a specified gamut to reproduce visual objects. Now, the hypothesis here. I've said hypothesis like a million times so far, but this is another one. A hypothesis here is that it should be possible to stimulate different mechanosensory neurons in the skin using a materials, and in most cases a nanomaterials style, nanomaterials approach in the right combination to simulate sensations like sticky, slimy, cold, smooth, high and low, or cold, hot, rough, smooth, high and low thermal conductivity, and so forth, using materials that can reconfigure themselves on the molecular, nano, or microstructural scale. So is there such thing as the Rgb of touch? So this illustration on the right-hand side is really not something we've done yet, not something that exists yet, but imagine that you can reach your hand into a box in virtual reality, and you can feel around for objects that differ on the basis of surface texture, or maybe this is a hot stone, so you could differentiate by temperature or mechanical properties, and this is a basketball, which could be squishy to some extent. So is there some array of tactile actuators that stimulate the mechanosensory neurons to achieve these sensations? So what we're basically proposing is to have sensors and actuators on a device. So the sensors communicate with some system and then send a signal back to the wearer of some sensation. And it's mediated by either an object in virtual reality, so you control an object in VR, or you control like a robot hand or something. The robot hand could have sensors on it and send tactile information back. So these are the results of an electrical engineering master's project. And this is my student who's controlling this robot hand. And the robot hand is outfitted with pressure sensors and temperature sensors. And the glove has vibrotactile actuators and thermoelectric devices. What's a thermoelectric device? It's a device that it's a semiconductor device that becomes either hot or cool depending on the polarity of the voltage you apply to it. So in principle, if you reach into a virtual environment, or even you have a robot hand that you're controlling, reach into a refrigerator and grab for a can of Coke, and you can feel the slipperiness due to the condensation, the physical resistance of the object, and also the temperature. Now, you'll just have to trust me that when the student pressed on the actuators, or the sensors here, the actuators here lit up, or in this case vibrated, you can't really see that on the screen, although in the future maybe we'll have what Aldous Huxley in Brave New World called the feelies, where you put your hands on these globes on the, like right where the cup holders would be in the movie theater, and you actually feel what the actors on the screen are feeling. We don't have that yet, but maybe if we get some funding for this project, we could have that. So there are example applications in VR, AR, telesurgery, training, teaching, physical therapy. But what we hope to do is develop some design rules, or at least some rough criteria for electronic skin, prosthetics, and robotics. The goal for material science, and I would say nanoengineering in particular, is to have spatial control. So all of these devices here are kind of like Radio Shack style devices that you could buy off the shelf. But what if we have spatial control, then each finger might be able to get the sense of translation of a surface, even if the interface is static. Skin conformability. So, for example, if you could do this with a surgical glove and really enhance the sensations, and what I think to get a greater range of sensation that is now available, what is now available, basically things that feel like tingling and like indiscriminate shocking and shaking. What we need are molecular or nanomaterials. So we started out really simply by trying to figure out if human beings had the ability to differentiate surfaces by a molecular monolayer of about 1 nanometer thickness on the surface. Now, this would be hydrophilic and hydrophobic substrates. So this is an oxidized silicon wafer, and this is a wafer coated with FOTS, which is basically a monolayer of Teflon. That's how you can think about it. It's only about 1 nanometer thick, and you can see that you get this wetting behavior, which shows that it's a very low surface energy. This is a very high surface energy. So the question is, can human subjects touch these objects and tell which is which? And the question is, how can they do it, if they can? And we worked with V.S. Ramachandran in our psychology department, who's actually a trained physician who's now more known for his contributions to cognitive science, in particular the phantom limbs and treatment for phantom limb pain, and also did some of the pioneering work in synesthesia. Now, synesthetes are people whose senses are mixed, where they can, for example, I'm making this up, touch a tabletop and taste peanut butter at the same time. That kind of thing. See a number 7 and always perceive it as being red. That kind of thing. So as engineers, we're not used to working with human subject data, but working with psychologists, at least you have a chance to get some meaningful data. So we used what's called an odd man out. One of these things is not like the other methodology, where you give the subject 3 different surfaces and one of them is different from the other 2. And you do some statistical analysis, and the odds of getting this right by accident are 33%, and it turns out that human beings are pretty good at doing this. Not great, but pretty good. Now, you might remember from earlier on in the course, we were talking about the differences, like in gecko adhesion, between van der Waals adhesion and capillary adhesion. So it's possible that the subjects were feeling contributions to capillary effects. Now, how do you eliminate capillary effects? You immerse the whole thing in water, because then you get rid of the meniscus. Turns out if you immerse it in water, humans are actually better at determining the differences. So it looks like it's primarily an effect of van der Waals force. Now, why do they do better? We're not exactly sure. Our intuition would say they should do worse, not only because of the absence of capillary effects, but also because in a dielectric environment, you reduce dipolar interactions between the surface and the finger. So we're not entirely sure why this is. There might be a training effect because the subjects did the wet experiment after the dry experiment, and they might have known what to look for. Now, it's also true that when you engage with the surface, it's not just the sliding friction, but also the articulation, the onset and release of the contact. Now, This is something that I read in a newspaper article. I'm not totally sure it's true, but if you have an instrument, if you're listening to a musical instrument like a trombone or an oboe, if they play exactly the same pitch, it becomes much more difficult to tell which instrument is which if you cut off the articulation. So if you cut off that beginning sound, it's actually most instruments sound the same, which I find is incredible, or at least human beings' perception of difference is significantly reduced. So anyway, we thought something similar might be happening here. So if you eliminate the sliding friction and you just say you can tap only, tell the subjects you can only tap, then they still do pretty well, although not so well as if they're allowed to slide. It turns out that the proportion correct actually decreases with the sweatiness of their hands. So I probably wouldn't do so well on this exam, especially now talking and doing some public speaking. So as the skin moisture increases, the proportion correct goes down. In fact, they become worse than chance here. So you know, the answer to the question, can we distinguish hydrophilic and hydrophobic surfaces that differ by 1 molecule thick? The answer is yes. And how? By the vibrations produced by different stick-slip friction behavior. Now, it's possible to determine the difference by tapping, but not as easy, and increased skin moisture makes discrimination more difficult. Now, we could do something kind of cool with this, which is to decode information by touch. And so you can use, something called the Ascii alphabet is what's used to encode characters in a computer. So if you have patches of hydrophobic and hydrophilic monolayers in 8 bits long, where your hydrophilic is a 1 and your hydrophobic is a 0, and you ask the subjects to scan their fingers along, after about 4.5 minutes of doing this, most of them can actually decode the word. In this case, it's capital LAB. Now what's interesting is that because we use the Ascii alphabet, we're actually case sensitive. What's not so cool here is how long it took. So a good internet connection might be like 100 megabits per second. Now we were able to do this at a rate of about 100 millibits per second. So about 10 seconds to get to 1 bit. So we're not going to compete with hard drive storage, at least for a while, until, I don't know, CRISPR increases our ability to do this. Okay, so this is like, it's not anything like Braille really, but it's kind of akin to Braille in the sense that you could, in principle, encode information in surface topography. Now, I say topography. Aren't these atomically flat? Well, almost. They're almost atomically flat. They have the same roughness within error as measured by atomic force microscopy. So what is actually the difference here? And the difference is between these surfaces, we're postulating is a difference in stick-slip friction behavior. Now, what's stick-slip friction? Stick-slip friction is the friction that is responsible for buildup of stress and then release, buildup of stress and then release, and then over several cycles, you end up getting like a tone, like a violin on like a violin bow on a string. That's actually created by stick-slip friction. Same thing with a rusty door hinge. So we can make a mock finger. This work was done by Charles Dong, who's a postdoc in my group. So you drag this mock finger that's made of silicone rubber with the tack removed by a brief oxidation. So it feels, it's hard to reproduce a finger, but as close as we could get with materials that we could make in the lab. And we generated these friction force traces. So we did 3 pulls. So and this is what the force looks like. Now you could imagine that if you zoom in to this friction trace, and if it makes like a vibration, so if it's making a sound, it's making a vibration, although we couldn't hear it, but it is making a vibration, and you compare the vibrations produced by the finger on a hydrophilic surface versus a hydrophobic surface, you could imagine that if they were exactly the same, they would look like this. If they were different, they might, look like this. And we can quantify the amount of dissimilarity using a correlational analysis. So these are, for example, using a velocity of 2.5 millimeters per second at no added weight beyond the weight of the finger. You see that these are actually quite different. And if you do this correlational analysis, you can see that there's a lot of skew between the types of traces. Now these ones are a little more similar. So see, we've got the spike here, and the periodicity is a little bit similar. This trial here was almost exactly the same, and you get more symmetry in this correlational analysis. Now you can also model the friction. So lots of different friction models have mathematically been conceived of, and if you plug in the parameters that are closest to the experimental case, you can get friction traces that are different or very similar. Now it's possible to take these individual trials and express them in a two-dimensional matrix where the two axes are loading force on one axis and scanning velocity on the other axis. And this is what the discriminability matrices look like. So we have the correlation, which is the amount different, kind of like the area under the curves, how are those different? And then the skew, that is how asymmetric are the different curves. And if you combine them together, you get a matrix like this. You've got velocity versus mass or loading force. And in some regions, we have more discriminability. So green and yellow, that means that the friction traces are more different, which tells us, and these here are the results of the mathematical model, which have this kind of high to the left, low to the right discriminability. So here, where it's red, they're the same. Here, where it's green and yellow, they're different. So if you want to make surfaces that are different, you want to be here. Now, what's interesting is that the human subjects, when they did this experiment, didn't construct a matrix in their mind. They just did it. They kind of automatically homed in on the right combinations of velocity and force that permitted the discriminability, which is pretty cool. It's like human machine learning, or just learning. Interestingly, if you add fingerprints to the system, so you take the elastomeric slab and you put ridges in it, the same periodicity as fingerprints, actually the discriminability goes up. Now, I'm not a biologist, but there are some competing theories that say, are the purposes of fingerprints due to, do they enhance grip, or do they enhance the tactile sense? There's some credibility that they might enhance the tactile sense due to roughness, but they might also enhance the tactile sense, maybe, due to differences in surface energy. At least that's what this mechanical model suggests. Now, this slide is highly speculative, but I want to throw it out here anyway. The surface of the skin, the glabrous skin, that is the side of your hand that has no hair on it, has mechanoreceptors, 4 different types of mechanoreceptors that differ on the basis of how quickly they react and how far deep they are in the skin, which affects something called their receptive field. The receptive field is just the area covered by each mechanoreceptor, even if the mechanoreceptor is very small, due to skin stretching and tissue deformation and so on. And what happens when one of these nerve endings is perturbed? What happens is that there's an exchange of sodium and potassium. So sodium goes into the cell, and then you generate what's called an action potential, which is an electrical signal which propagates up the neuron to the ganglion of cell bodies in the spinal cord, and then up into the brain where it's perceived as an object in consciousness. So is it possible, because these different mechanoreceptors have different frequencies that they're responsible for detecting, to use our analysis to narrow down which biological structure is responding to which type of force? So these are the four different mechanoreceptors in glabrous or hairless skin, and this is hairy skin, which the only difference here, it still has these, but it also has hair follicle receptors as well. Okay, so this is purely a computational model that shows a model finger with these different zones that represent fingerprint ridges, and you get these plots of force, and they're plotted at each position. So So what happens is that this is kind of like a cascade. If you were to look at a video of this, it would be like kind of like this. Does that make sense? So say these are fingerprints, and as you drag the silicone slab mock finger across the surface, you get these oscillations, which are kind of in sync, but sort of die down at the end, at the back end. In some cases, you get oscillations. These are kind of rare events where they propagate in reverse. It's actually like if you were to take a piece of silly putty and you were to put it on a piece of glass and drag it across, you would kind of, and put it in slow motion, you would kind of see these sort of vibrations. I shouldn't gesticulate quite so much. Okay, you would sort of see these vibrations from underneath. Anyway, you can deconvolute these master plots into different frequency ranges, and then it's possible actually to do a cross-correlational analysis on these different ranges and score which frequency ranges are you going to find the biggest differences. And the biggest differences in frequencies between hydrophilic and hydrophobic surfaces might, highly speculative, correspond to the biological structures that are most able to detect that kind of interaction with a surface. So this is all computational, and really to test this hypothesis, you would need to innervate with a probe and correlate the action potentials generated with the vibrational frequencies that you're creating using touch. So it would be a really complicated experiment, but this is how science works. This is an observation, then you would do the hypothesis. So in this example, maybe this one has low correlation and high discriminability. These ones are too slow to respond to high fine texture, so you throw those away. This one is high correlation, low discriminability, and this one might win. That would be the Meissner's corpuscle, and conveniently, the Meissner's corpuscles are kind of have a distribution a distribution which is commensurate with the pitch of the fingerprints. So maybe. All right. Another area that we were interested in was, and this was the credit for this project also goes to my postdoc Charles Dong, is the perception of softness. So in engineering sciences, softness is kind of like the opposite of hardness. And hardness can be measured using a variety of tests. So you take an indenter and you plunge it into a material and you measure how far into the material it goes and how wide the indentation spreads out, and that's how you get the hardness, roughly. How does a human being determine hardness? Or softness. So, okay, this is kind of hard, but a diamond is harder. This chair is a little softer, but still pretty hard. Like, what are we doing physiologically, or what are we doing perceptually or cognitively to determine what is soft, and what mechanical cues are we actually looking for? What materials properties do those mechanical cues correspond to? So the parameters that we're primarily testing in an indentation test in a lab are contact area contact area and indentation depth. And for most isotropic, that is unstructured materials that have bulk dimensions, when you press down into it, the contact area and the indentation depth increase at the same rate. So it's hard to decouple these effects. But in real materials that have anisotropic structures and are porous at the surface, you might get some other behavior. So the goal here, so this is a diagram that shows the force. This delta here is the indentation depth. This is the contact radius, so pi a squared would be the contact area. And the goal is to manipulate the contact area and indentation depth independently by creating these micro or possibly someday even nano pores in the surface to the extent that that might help test a hypothesis, and then create an array of these rubber slabs and try to differentiate the two types of tactile cues. So the contact area is modulated by micropatterning the surfaces with photolithography, and the indentation depth is modulated using the slab thickness. Now, why the thickness and not the modulus? Because we wanted to avoid a scenario in which the bulk softness of the material determined our perception of softness. You can make something that's soft feel really hard if it's thin. If it's thin and you try to press into it, the deformation is limited because the deformation is pinned at the bottom surface, and it can't deform enough to feel soft. So instead of modulating the modulus, that is the intrinsic stiffness of the material, we're modulating the thickness, and that's how we're controlling it. In fact, we created these slabs using random values of elastic modulus just to prove the point that it wasn't just modulus that was creating the sense of softness. Okay, now this this paper, which is in revision right now, I've never talked about in front of an audience, so I haven't focus group tested any of my explanations, so forgive me, but I'll do the best I can. So Pitzer posts are created in slabs to determine the contact area, and the force of the human finger is calibrated using a technique that Kenneth Lowe in structural engineering, Professor Lowe's group, is working on, and that's called electrical impedance tomography. So here are some microscope images of the posts or wells, and then these are results of electrical impedance tomography. Basically, this is an electrical trampoline. And wherever the little kid or grown adult is standing on the trampoline, it lights up. Because the impedance changes here. So this is a human finger and an acrylic indenter. So we're normalizing. We're trying to make sure that we get the right range of forces that human subjects are actually applying to these surfaces. And here's a table, and I want you to memorize every element. No, just kidding. So this is actually it's a really, sometimes you get a really complicated data set. And it's the biggest challenge in the world is to figure out how you're going to describe it when each data point contains like modulus information, indentation depth information, and contact area information. So the best we could do here is pick these parameters and then give them circles, squares, or triangles, and then label them with blue, green, and red. Okay, you don't really have to, I'll point out the trends. Okay, so we use two different psychophysical methods with human subjects. One was a two alternative force choice test, where we give you two samples, and we do this over and over again, and say which one's softer, these two, which one's softer, these two, which one's softer. It's like at the eye doctor, better one, better two, better one, better two. And sometimes they give you the same one twice, but they say better one, better two, better three, and better four. But better three is like exactly the same as better one, and it's the same test, but they're trying to mess with you. But anyway, that's what we did to these people. So these are the scatter plots, and they only really make sense if you look at the color coding here. So as you increase the indentation depth, they are more likely to be perceived as softer. Okay, doesn't really offend the intuition too much. Also, as you increase the contact area, you also tend to increase the perception of softer, except for the triangles, which are all pretty, which tend to go down. So there might be a competing effect here. Now, if you plot these data instead as a function of the, and you put them in order, light red, light blue, red, light green, and so on, you get this, this trend here. Now, if you plot this kind of matrix where you're comparing each one of the slabs to each other, so this is like the competition of like which superhero is going to beat which other superhero, like Superman and Silver Surfer, or Wonder Woman and Rogue, or however it might be. And this shows that we're more or less constant in saying that light green is going to beat out light red, depending on this scale using this test. Psychophysical method 2 was the magnitude estimation test, where you give the subjects all of the samples and they have to place them on an ordinal scale, like Mohs hardness. So like you might have remembered this from like earth science in middle school, where you have corundum versus silica versus diamond, and you can place them all on a scale. And that's what this test is akin to. And here's what it looks like. So pretty good. There are 5 subjects each. Generally, they're clustered around the similar values. We can do a similar type of analysis here with the matrix, and then we can plot the relative hardness, and we can see that the magnitude estimation test and the 2 alternative force choice tests gave essentially the same results, with a little bit of swapping out here. Now this analysis here is the combination of some really sophisticated statistical methods, which I'm not going to go into to the extent that I'm not really prepared to go into it. But basically, the lower on this curve, the better the mechanical model is at determining which material is softer. So it turns out that some combination of adding the contact area with the square root of the indentation depth gives you the best chance to engineer something that's soft. Okay, now how is this useful? If you have a virtual object, or an object that can reconfigure its properties in real time, and you want to say, I need this to feel soft like an orange, then you press a button or have a computer algorithm change the properties of the material such that it has the right combination of contact area and indentation depth to dial in that perception of softness. That's why combining the mechanical model, getting the accurate mechanical model, which reproduces most closely the human subject perception data, is the key to engineering a human-machine interface. All right, just 2 little vignettes, and then we'll have a discussion if you want. So P.PSS is a conductive polymer that we would like to be able to use in devices like, say, electrotactile devices. Electrotactile devices are devices which apply an alternating electric field to the skin in order to stimulate action potentials in the mechanoreceptors without actually pressing on an object. Now, in its native form, P.PSS, which is just this material, so this is the conductive part, and then this is kind of the scaffold for the material, polystyrene sulfonate. it's very mechanically brittle. So what my postdoc Laura Kaiser did was make this new type of scaffold that had this bottle brush segment. So you can see sort of why it's called a bottle brush. And a bottle brush is kind of mimics the proteoglycans in your joints, which give them mechanical give, and they're pretty mechanically flexible. So here's some chemistry. You can ignore all of it, and here is the stuff that you get. And if you buy this stuff in a catalog, it looks just like this. So by eyeball spectrometer, we have something that should be conductive, and it is. It's also touch healing, so you can place the two pieces together, and they'll kind of reform, and it's quite stretchable. So this is a stretchable conductor that we might be able to use in haptic devices. We did a little bit of work with ionic conductors. So signal transduction in electronics is done with electrons, but much of signal transduction in biology is mediated by flows of ion currents. So can you create a bio-ionic interface if you use conductive Ayano gels? So these are hydrogels where you have put in like a saturated sodium chloride solution. Now, not only do you need to do that, because like a hydrogel, like a contact lens, if you put it out on a surface, it will dry out. But if you swap out the water for like glycerol, then the vapor pressure is way lower and it won't dry out. So here's our device that has these little Ayano gel pixels in it. And here's our Ayano gel thimble, and here's the sensation curve where we have the voltage versus the frequency. And the bottom curve here is where the subjects can just feel the sensation, and up here it is uncomfortable. And then we're saying that the top of the error bars here is kind of the sweet spot. That's where you want to be. All right, so the final thing. Kinesthesia, it's different from the tactile sense, but it uses some of the same biological structures, so that is the sense of motion. So current kinesthetic devices are fast, but they're bulky. So if you've ever had one of these VR experiences where you put your hands in these stirrups and it's a big bulky machine, but you can kind of get an idea of physical objects on the screen. What we thought was using a polymer that had a thermoplastic polymer that had a glass transition temperature that was close to the skin temperature. If you modulate it up, the temperature up and down to above and below the glass transition temperature, you should be able to like freeze the textile, so freeze the glove in position to mimic the movement of an object, or the resistance of an object. So this is a sensor glove that has thermoelectric devices on it, which either heat up or cool down. And we connected it to this robotic finger. When we bent our finger, the robotic finger bends up, hits this acrylic wall, it hits a pressure sensor on the robotic finger, which sends a signal to the thermoelectric device to cool down and to freeze the thermoplastic polymer. So our results here, it takes kind of a long time. It's like a 10-20 second response time, but it's never been done before. But the cool thing about it is that if you're a little overzealous and you like crunch the glove in its frozen state and you break all that material, you can put it in your easy bake oven and it self-heals, so it reforms across the fractured interfaces. So much of this work was done by my students, Cody Carpenter, Charles Dong. Nick Root is a student in V.S. Ramachandran's group in psychology, and funded by the Center for Wearable Sensors and also the NIH New Innovator Award. And we have a few minutes for questions, actually like 3 minutes for questions, which I'm happy to take.